All right, we can get started. So hello everyone, and welcome to this year's iteration of our Stem Cell Research Conference, ReGen21, Regenerating the Science Network, brought to you by the University of Toronto's Student Society for Stem Cell Research. My name is Jacob, and I am the conference co-chair alongside my fellow co-chair Bapasha, and we are beyond ecstatic to bring this conference to you all today. The past year has undoubtedly been a long series of ups and downs, and so before we get started, we would like to extend our gratitude to our amazing team of executives that have worked so tirelessly in transitioning our annual conference to the virtual delivery format that you see this year. Furthermore, this conference could not have been possible without our amazing sponsors, so I'd like to thank the Student Initiative Fund, our gold sponsors Genome Canada, MD Consultants, and CCRM, our silver sponsors, the U of T Cells and System Biology Department and Stem Cell Network, and our bronze sponsor, WeStem Academy. Now more than ever, new scientific developments and research are at the forefront of attention, and our hope is that this conference will imbue the discoveries and progress in stem cell research with connections to ethics, policy, innovation, and technology. Before we get started, we wanted to provide a brief overview of our organization, the Student Society for Stem Cell Research, or also known as SSSCR. We are a multinational organization that focuses on the advancement of stem cell research and awareness regarding its potential. The mission of our organization, put simply, is to empower our generation to fulfill the hope and promise of regenerative medicine through stem cell research in order to alleviate human suffering. More specifically, we here at the U of T chapter of SSSCR aims to raise awareness and further educate our audience of the potential of stem cell research and regenerative medicine. We strive to accomplish our mission through three primary objectives. Firstly, we aim to provide information and educate both younger and older generations about the promise of stem cell research. Second, our endeavors are designed to advocate and inspire interest in stem cell research in our community. And lastly, we task ourselves with creating a network of individuals through which we can facilitate and expedite the process of stem cell research.
Events such as this conference allow us to tackle these three facets of SSSCR's mission. Through our conference, we aim to inform, inspire, and connect participants through a variety of workshops, seminars, and competitions hosted by experts in this field. Taking advantage of this year's virtual format, we are hosting speakers and students from all over the world. This year, we hope to highlight the interdisciplinarity of the field of stem cells to include unique viewpoints from not only scientific research, but also business, law, and ethics. To this end, we have divided our conference into three main Zoom webinar halls: Research, Bioethics, and Law and Business. To start off, we are presently in the Research Hall, where after this opening ceremony, the keynote speech by Dr. Derrick Rossi and a special moderated Q&A with Dr. James Till will take place. This will be followed by live presentations and judging for the Research and Bioethics case competitions in the Research and Bioethics halls, respectively. We encourage all, even non-participants, to attend and support the teams who have been working tirelessly over the past few weeks on their proposals. Then the conference will be divided into the three halls, each featuring 3 consecutive seminar presentations and a formal panel discussion. After this, we will reconvene in the Bioethics Hall, where a lively debate on stem cell bioethics between Dr. Megan Muncy and Dr. Charles Binkley will take place. We will then end with our closing speaker, Dr. Michael Laflamme, and final remarks from the team. Also, we are excited to announce that our poster gallery is now open. Head on to the competitions tab of our website whenever you get the chance to view research done by our attendees. The day sure is packed with many exciting talks and networking opportunities, and we hope you all are as excited as we are to learn from and connect with fellow stem cell enthusiasts. With this, I see no better way to kick off our conference than by the opening remarks from our first speaker, Dr. Arnold Kaplan, who we will now promote to panelists, so please be ready to share your audio and video. Dr. Arnold Kaplan is a current professor of biology at the Case Western Reserve University in Cleveland, Ohio, and the current director at the university's Skeletal Research Center. Known as the father of mesenchymal stem cells, or MSCs, Dr. Kaplan is the scientist who first popularized MSCs. Dr. Kaplan's research investigates how MSCs are able to recover and improve damaged cells and tissues. Thank you for being here, Dr. Kaplan, and I'll now hand the floor over to you.
Can you see my screen?
Yes.
Can you see me?
No, not yet.
Okay, well, somebody needs to put my camera on because I really look pretty.
There's the share video option at the bottom of your screen. Do you see it?
Yeah, it doesn't show up, but I will forget about that and start my, oh, I see it. Okay, now I see
Excellent.
it.
It's good to see
Okay,
you.
so now you can see me.
Yes.
Okay, so first of all, it's a huge honor to start these proceedings. And I'm grateful for the organizers, and I'm delighted to take part in these proceedings. So these are my obligatory disclosures. I started a company called Osiris Therapeutics. I have no association with Osiris. The university receives royalties, which they generously share with me. You can rest assured I'm not going to retire on these royalties. This is actually the key slide for my entire talk. And this is a page from my histology textbook when I was in medical school in 1963. And what you see here, and actually I'll try to use this laser pointer, you can see that these cells go through distinctive morphological changes and eventually can become erythrocytes or all of the blood cells. And this entire concept was documented by hematologists doing blood smears. There was no molecular biology in these early 60s, and you young people now would say that things back then were pretty crude, but these scientists put together what is now a lineage diagram of changes. And we know, for example, now that the molecular control elements that control this differentiation stage also actually affect this differentiation stage. So the uh, the marrow microenvironment hugely complicated, and yet with just simple blood smears, these guys deduced this whole lineage, and, and they proposed that there were stem cells, which we now call hemopoetic stem cells. So in the early 60s, and I'm focusing on James Till, who, by the way, everybody should send him a happy birthday on August 25th. He'll be 90 years old. It's quite a wonderful number. Well, Till and his colleague McCullough in the 60s lethally irradiated mice and then repopulated those mice with whole marrow. And they looked at the spleens. And in the spleen, they showed that there were lumps that they hypothesized were from single marrow cells, and they speculated that these were hemopoetic stem cells. And there's a Nature article which summarizes much of this speculation. So I'm going to talk about, very quickly in introductory fashion, the 60 years since the 1960s, because there are major accomplishments in the stem cell theory. And you're going to see, I'm going to challenge the use of these words stem cells, because I myself made a huge error in calling some cells stem cells, which were not. So in the 1960s, Dr. E. Donald Thomas in 1969 was the first person to do and publish a bone marrow transplant, which he used for a leukemia patient using match sieves. And in 1990, indeed, he received the Nobel Prize for this. Likewise, Boxmeyer, Boyce, and Auerbach showed that cord blood could also be used in successful transplants. And and they transplanted cord blood into a 6-year-old young boy who was suffering from Fanconi's anemia. So whole marrow, cord blood, all got their starts during this era. In 1990, James Thompson started actually the whole embryonic stem cell story. And in this case, he published a paper on human embryonic stem cells. And this really started the era of regenerative therapies that could be envisioned from this. I could say that in 1991, I wrote a paper and coined the term mesenchymal stem cells. And you'll see that I'll give you the basis for why I call these cells stem cells, and I'll give you the basis for why I think this was a huge mistake on my part. The whole stem cell era got gigantic publicity when President George Bush in the United States limited the research that could be done with embryonic stem cells. And so therefore, very creative companies, including Geron, which I'll talk about in a minute, were started to actually use embryonic stem cells and investigate their potential therapeutics for human use. I'd also like to say that in 2006, Dr. Yamanaka introduced us to induced pluripotent stem cells, iPS cells. And this actually countered all of the objections that people raised about embryonic stem cells, because we could identify and isolate pluripotent stem cells using the technologies that he and his colleagues first introduced. I want to say that in 2010, there were 100 clinics who advertised stem cell procedures. I'll come back to that in a few minutes. For me, year 2000 turned out to be a huge paradigm shift, because we understood now that MSCs actually could influence the immune system and produce a regenerative microenvironment, and indeed that in the body, MSCs are not stem cells. I'd also like to say that in 2011, Geron abandoned its stem cell therapy. It's been revived by commercial entities, but I'm not going to go into that. So I want to say, in the strongest terms, there are no stem cells in the adult body. This includes hemopoetic stem cells. These are committed progenitors. Hemopoetic stem cells cannot make neurons, cannot make muscle. They can make all of the blood cells, as I'll describe in a minute, but they're not pluripotent, they're multipotent, which means they're committed progenitors. You can still call it stem cells, but calling them stem cells has led to real problems in terms of therapies that are being promised by some practitioners. In 2010, I wrote an article and I said I made a mistake with MSCs, and I changed their name to medicinal signaling cells because they make drugs at sites of injury. I'll talk about that in a minute. So here is lineage map for lung, for liver, for brain, and this part of the diagram is for all of the cells in blood cells, and I've enlarged that, and this is the hemopoetic stem cell, which again is a committed progenitor. It can only go down these pathways. It's multipotent, not pluripotent, not a real stem cell, but it gives rise to all of these blood cells. And so in that case, it can be loosely called a stem cell, although you have to keep in your head this concept of committed progenitors and multipotency. So again, erythrocytes, B cells, T cells, macrophage, all derived from this cell. And this is a diagram which I drew in the late 1980s, and you can see that I proposed that in bone marrow, I could isolate a cell, which I called a mesenchymal stem cell, which I could push down a bone forming or cartilage forming or muscle forming or fat forming pathway. And I could indeed do all of these things in culture, and I could get this cell to divide in culture and keep its multipotency. And I made the simplified assumption, which was consistent with the science of the day, that this is what happens in the body. This is not what happens in the body. And the other important point of this diagram is you could see that I stole the logic of hemopoiesis and adapted it for mesengenesis. So again, this looks like the hemopoietic pathway, but these are mesodermal phenotypes. If you want to isolate MSCs from somebody's marrow and expand them, you can easily do tissue culture with that. So it's an in vitro technology which makes use of the ability to push these cells down these lineages. I'm not going to talk about that. But if you want to do regenerative medicine, you have to understand what the properties are of the MSC itself, because what does the MSC do in the body naturally, and how can we mimic that therapeutically? So all of the cells, neurostem cells, hemopoietic stem cells, liver stem cells, heart stem cells, are committed progenitors. They're multipotent. The MSC is not a committed progenitor in your body. All of the multipotency that we see is in tissue culture. So I can make cells dance on a tissue culture plate. This is not what they do in the body. So MSCs arise from perivascular cells. This is a capillary in heart. And if this vessel breaks, the pericyte comes off and it becomes an MSC. So again, injury to the blood vessel, pericyte comes off, differentiates, changes into an MSC, and the MSC senses its microenvironment at the site of injury, and it makes molecules that control the immune system, or it makes molecules that set up a regenerative microenvironment, anti-scarring, pain management, brings in new blood vessels, a regenerative microenvironment. And if you go to clinicaltrials.gov, you'll see there are over 1,200 clinical trials listed. I put all the clinical symptoms here. I put it in higher font so you could see Crohn's disease, MS, kidney transplant, acute myocardial infarct, rheumatoid arthritis. What do these have in common? These have in common immunomodulation, regeneration, and there's a whole separate activity that MSCs protect from the invasion of foreign elements, and I'll talk about that in a minute. Hemopoietic stem cell, again, committed progenitor. They're dominant, multipotent. They have a well-defined niche, which I'm going to show you in a few minutes. They actively sense and respond to where they're situated. They produce immunomodulatory molecules, and they are hugely secretory and regenerative with regard to the bloodstream. But all of those stem cells, all of those tissue-specific progenitors, they all can be characterized by this same descriptor. So I want to show this slide. It's one of my last slides from a textbook. And then they blow up marrow. And of course, in vitro, we can take an MSC and we can get it to go down all these lineage pathways. But I want to focus on the hemopoietic stem cell. This hemopoietic stem cell makes red blood cells, makes platelets, makes macrophage, makes B and T cells. Look at the way this is drawn. So this is the niche for the hemopoietic stem cell. And I put this into a diagram that I'll show you. The hemopoietic stem cell, or all of the tissue-specific stem cells, neurostem cells, liver stem cells, heart stem cells, are sitting in contact with their own very specific perivascular pre-MSC, and both of these cells are in contact with a blood vessel and with circulating blood. And this cell is producing niche specific molecules for this progenitor. This is in every one of these tissues, the story is the same. So there is a universal, and again, these are committed progenitors, stem cell niche. This is Diego Correa and Rodrigo Somoza, and they helped me put together this Nature poster, which I suggest you go to this website and you can download free because it has an enormous amount of information about MSCs. I'm going to highlight this. Again, I talked about this universal stem cell niche, which is described in the poster, but I want to focus on active hemopoietic stem cells making progenitors, and there's quiescent hemopoietic progenitors. So these hemopoietic progenitors are sitting on their own unique MSC perisite, and these cells are making niche specific molecules. So they're making molecules that make this cell quiet and this cell active, and so therefore these two cells have different signatures, and we can sort them out of marrow and show you that they're molecularly different, and by single cell RNA-seq, they make different molecules. One last point, every textbook, medical textbook, says that blood vessels are controlled by sympathetic neurons. The contractile element for blood vessels is the perisite. So when I took my blood vessel medication, my blood pressure medication this morning, it affects the perisite, which can squeeze or relax around the blood vessel. And likewise, sympathetic innervation is to pericytes, not to blood vessel cells. So again, download this poster, huge amount of information about this particular stem cell. So MSCs come from perivascular locations. They survey, sense, and respond to their local environment. They produce immunomodulation, both anti-inflammatory and pro-inflammatory. If you have bacteria, you want pro-inflammatory to bring in macrophage and monocytes to eat those bacteria. They're immunoevasive, which means you can use allogeneic MSCs for therapeutic purposes. And the mind blower, the true mind blower for me, is that MSCs at sites of injury make molecules that sit on opioid receptor and manage pain. They make mitogens for those tissue intrinsic stem cells. And again, MSCs themselves are not stem cells. And last, MSCs secrete proteins that kill bacteria on contact. That's why young women can have monthly bleeds and never have sepsis, because if as they break all those blood vessels, they release MSCs. If an MSC sees a bacteria, it makes molecules that kill the bacteria. These molecules, it turns out we now know, are antiviral, and there are now clinical trials that appear to indicate that MSCs can be curative for COVID-19 infections. And there's published clinical trials that I urge you to go look up and read. It's another mind blower for all of us. So if you see an ad for stem cells and a stem cell product, there are no stem cells which are approved by any regulatory agency. There are no stem cells that are pluripotent that can provide therapeutic effect. So these are bad actors, And that's why I changed the name of mesenchymal stem cells, because these bad actors were using the stem cell terminology to fleece patients, because there are no stem cell therapies. There's MSC therapies, there are hemopoetic stem cell therapies, there are no pluripotent stem cell therapies. I give this course every year. Go to this website and download the course. I invite incredible people to talk about a variety of stem cells, cell-based therapy, and tissue engineering. There's clearly a lot of people at the Skeletal Research Center who assist me in all of the science I do, and I'm indebted to them and to Case Western Reserve for everything they do to assist me. So again, I look forward to the rest of this conference, and I'm happy to participate.
Thank you so much, Dr. Kaplan, for your insightful introductory remarks. I think that was a great introduction to the field and concept of stem cells, and we're really grateful for it. Unfortunately, we don't have time for any more questions, so we will now redesignate you to attendee status, and we really hope that you enjoy the rest of the conference and continue to participate. Thank you once again.
Again, thanks for inviting me.
For sure, thank you. I would now like to invite our keynote speaker, Dr. Derrick Rossi, to the stage, who we will now promote to panelists. So please be ready to share your audio and video. Dr. Derrick Rossi has served as a professor in the Harvard Department of Stem Cell and Regenerative Biology, as an investigator at the Immune Disease Institute in Children's Hospital, Boston, and as the founder of biotechnology companies Moderna, one of the leading developers of the COVID-19 vaccines, Magenta Therapeutics, and Intellia Therapeutics. During his time as an investigator, Dr. Rossi's research has primarily focused on the fundamental mechanisms of self-renewal and multipotency in hematopoietic stem cells, the aging of HSCs, and stem cell reprogramming. For his research work, Dr. Rossi has received numerous awards and even been featured on Time magazine. We're super excited to have you, Dr. Rossi, and look forward to your talk. The floor is yours.
Okay, thank you. Hello, everybody. Can you hear me?
Yes, we can, and we can see you as well.
All right, let me try to share my screen there. Then let's see, I'll do this one. And can you see my screen?
Yes.
Okay, and then let me get a laser pointer here. Okay, should be good. Good morning, everybody. Happy to be with you today, my alma mater, the University of Toronto. So today I'm going to talk to you about my journey in stem cell science and some new therapeutic strategies that have emerged from some of the things that we've done in the lab, we and others, of course. So I'm going to take you through a few different science stories that went on to become biotech companies, one of which you're very familiar with, I'm sure. I was an associate professor at Harvard for many years, and I used to teach a course to undergrads that was focused on, moving one science from the bench to the bedside through the founding of biotech companies. And I challenged my undergrads to come up with virtual biotech companies. And for the course, I used to say, enjoy taking my course because I would invite all kinds of guest speakers in, venture capitalists, serial entrepreneurs, IP experts. I had a guy from the FDA fly in every year. So it really was a great way for me to do the course. But one thing I found interesting, and the students really got a kick out of, was that often the journey that each of these professionals took to get to where they ended up was not a straight line. It was so they would often go back into their story and how they ended up with where they were. So I thought I'd start off by telling you that story for myself, and it's a global story, but it starts at where most of you are right now at Toronto. I was born and bred, and I did an undergrad at U of T, and then I took a gap year after my undergrad. I went to Central Africa and hitchhiked across Africa and took a dugout canoe down the Congo, went back to Toronto and entered a PhD program working with Alan Bernstein, then at Mount Sinai Research Institute. But this is going to be a theme that repeats. I didn't actually finish my PhD there. Rather, I got a master's degree, and I instead flew across the pond again to Paris, live in Paris, where I entered yet another PhD program with a fellow named Olivier Delatte and Gilles Thomas. But it's another PhD that I didn't finish because I then moved to Dallas, where I worked with Dr. Mark Henkemeyer, who I actually first met in Toronto, where he was a postdoc working with Tony Pawson. Went to work with Mark for a year in Dallas. Flew again across the pond to Helsinki, Finland, where I entered yet a third PhD program working with Tomi Mäkelä. And I know you're all expecting a red X to appear on that PhD. So sorry to disappoint you, that one I actually finished. Also met my lab partner who became my wife. Did my postdoc at Stanford, lived in San Francisco, and I worked with a really great stem cell biologist, Irv Weissman, and started to have a family. Our first daughter was born as a fully developed 10-year-old child, oddly. Took my faculty position at Harvard in 2007, Harvard Medical School. Had a great lab there, which I ran until I retired from academia in 2018, and had a couple of other girls. So the conclusion, and I'll just end there and take questions, just kidding, travel the world, do some cool science, and work with some smart people. So let's maybe have a family along the way. So let's get on to the science, shall we? So I'm going to tell you about a workaround that my lab did that eventually led to this field we now know as mRNA therapeutics. And it starts in a great story in stem cell biology published by Shinya Yamanaka, shown here, who in 2006 demonstrated most elegantly that ectopic expression of 4 transcription factors in any somatic cell could reprogram their fate back to a pluripotent state, much like an embryonic stem cell, induced pluripotent stem cells. He, of course, wins the Nobel Prize 6 years later for this amazing discovery. So we were inspired by it at the time. Remember, I started my lab in 2007. Yamanaka had published, and I had just seen his work presented for the first time at a conference. I actually think it was in Toronto, the SSSCR conference, although it might have been San Francisco. And so we just, at Yamanaka, when he achieved his goal, he used a delivery vehicle, retroviruses. Actually, viruses come up multiple times today, you'll see. But he used retroviruses to deliver the cargo, these 4 transcription factors, but it had a problem. It has a problem that retroviruses integrate into the DNA, and some of these factors were quite serious proto-oncogenes, c-Myc, for example. And many in the field at the time were trying to figure out ways of eliminating this DNA footprint, this integration thing. So we thought we'd get into that as well. We were inspired. So one kind of a side project in the lab, we thought, well, let's just skip the DNA altogether. We know that DNA genes encode for mRNA in the nucleus, which migrate out to make proteins in the cytoplasm at the ribosome. So why don't we just skip the DNA, we'll use mRNA for reprogramming. Simple enough idea. But you have to wonder, mRNA was known to be the intermediate between DNA and proteins since the 1960s, early 1960s, actually. And, I think there are many molecular biologists watching this, and I bet you all use DNA on a regular basis in your labs, and you all use proteins on a regular basis, but how many of you use mRNA? My guess is not many. And that's a little odd, isn't it? Molecular biologists use anything in their toolkit that they possibly can, yet mRNA was not being used. So we thought, well, we were an mRNA biologists. We thought, let's just try it. So we made, classic textbook mRNAs, really very beautiful with all the regulatory sequences that one would need. And we introduced it into cells, and it didn't work. And it didn't work in spectacular fashion. We mostly achieved a lot of cell death in the dish. And of course, this is not what we were after. And the reason that we were tripping this sort of cell death, this apoptotic program, is sort of illustrated in this slide here. And it really comes from the fact that ever since, again, viruses, second mention, ever since viruses and cells first met one another, viruses have been trying to figure out ways of getting their nucleic acid into cells, and cells have been coming up with pathways and mechanisms to detect that, to deter that. So sure enough, as we were introducing single-stranded RNAs, mRNAs, they were going in via the endosome, trigger TLR7, leading to an NF-kappa B mediated interferon response. And we determined that this is what was happening with us. And by the way, if this sort of antiviral response is robust enough, it'll lead to cell death. So that could have been the end of the story, but we thought there might be a workaround. Well, as it turns out, nucleosides, which are the building blocks of RNAs and DNAs, actually RNA nucleosides are modified heavily in all the cells of our body. There's actually over 110 different modified nucleosides. And very importantly, work by two researchers at UPenn, Kathleen Carrico and Drew Weissman. They weren't actually working with mRNA, they were working with just sort of studying this antiviral response, little short stretches of RNA or longer stretches of RNA, but they had shown in a paper published in 2005 that certain modified nucleosides could abrogate this antiviral response. So we thought, well, maybe we can apply those modified nucleosides to mRNA synthesis, and we'll see if we can slip by these antiviral responses. So test protein first made, GFP mRNA for green fluorescent protein. So we transfected unmodified, textbook mRNA. You got a few green cells, but you've got a lot of cell death. Here's viability here. However, when we swapped out cytosine for using one of these modified nucleosides, 5-methylcytidine, now we got actually a lot of much greater viability and more green cells. Similarly, when we replaced all of uridine with pseudouridine, again, we really didn't kill all our cells and got a robust green signal. And because these are modifications on different nucleosides, we combined them, 5-methylcytidine and pseudouridine, and lo and behold, we now had very, very good viability and very, very robust expression. So we called these new mRNAs modified RNAs, mod RNAs for short. And then everybody in the lab started working on them for all of their favorite proteins. Now we had a tool that we could express anything we want. So, and they had great properties. So for example, here, this is a FACS plot showing, again, just the GFP protein. The more mRNA you put, the more expression you got. So it was dose titratable. That's a great property. Control how much you express. Important though, mRNA, it goes into the cell, it goes to the ribosome, it makes its protein, it's degraded. So this is a time course of GFP where, you put it in, you get a peak expression at around 12, 15 hours, but a few days later, depending upon the half-life of the protein, of course, you're back down to baseline. We found, however, though, that we could repeat dose because we had abrogated these antiviral responses. So here's an experiment showing that we dosed for 10 consecutive days with a modified RNA for GFP, and 10 days later, the cells are happily, this was a nuclear localized GFP, happily expressing GFP. We found that we could put in multiple different mRNAs at the same time and get them co-expressed. So here's a modified mRNA for cherry, here's one for GFP. We ended up making cocktails of 7 modified RNAs, get all the proteins expressed at the same time. We never went up to 25, but I'm sure that would have worked. And then we went back to our project at hand, which was directing cell fate with modified RNA. So the first experiment we did was a transdifferentiation. We put a MyoD modified mRNA into fibroblasts. We turned them into multinucleate kind of contractile myogenic cells. We, of course, succeeded in our goal at redirecting human fibroblasts back to pluripotency. It was actually incredibly robust. Essentially almost every cell in the dish was turned into an iPS cell in comparison to the traditional Yamanaka viral reprogramming. And an experiment we did in that study, which I thought was really important to address the elephant in the room, was, well, once we made these iPS cells, which we called our iPS cells, RNA, rest in peace, RNA iPS cells, we then used, again, another modified RNA to direct their fate to a myogenic lineage. So this study made a big splash in stem cell science. We got the cover of Cell Stem Cell, and had an artist sort of do an illustration, RNA directing cell fate. And it was popular in the lay press, written up all over. But it was really strange to me. I was getting calls from pharma, big pharma, on a kind of weekly basis saying, hey, we saw that study of yours. We're really interested in getting the license for that to make iPS cells. And I got multiple of these calls, and I thought, doesn't anybody actually recognize really what we've done here is now there's a new technology for expressing proteins. And nobody really sort of grasped that, which I found remarkable. But to me, DNA makes mRNA, makes protein, makes life. The ability to have your hand on mRNA to make protein really bespoke that it had a potential to translate for bigger things beyond stem cells. So if you were to envision modified mRNA as a therapeutic, what experimental evidence would you need to convince yourself that that is possible? Well, the first experiment we did made me believe it's possible. So here we made a modified mRNA encoding luciferase, firefly luciferase. It catalyzes luciferin substrate, light is emitted. Very first experiment we put into mice, we dosed into the thigh muscle, increasing concentrations of luciferase. We gave the mice IP injection of luciferin, the substrate, and put the animals in the IVIS box and saw that we were expressing luciferase in a dose-dependent manner in these animals. So this is the first experiment. First time we tried to put this in animals, it worked. That says something about the robustness. We did another experiment. I liked, therapeutic end. So I'm a blood stem cell biologist. Blood is my thing. So I thought, well, why not erythropoietin? We made a human erythropoietin. That's a cytokine that stimulates red blood cell production. It's the thing that athletes used to dope on to make them go faster and be stronger. We weren't trying to do that. We were trying to illustrate that we could have a therapeutic end. So we made a human erythropoietin, which is distinguishable from mouse. We could measure that it was being made in the mouse. We put it in the skeletal muscle again. It's a secreted protein. We could measure it in the blood. And then 10 days later, we could look at the blood cell counts and see a dose-dependent increase in red cell counts, hematocrit, and hemoglobin. So you could have a therapeutic end if you so wanted to increase red blood cell. That was just obviously a test case. We also started a project in the lab looking at vascular endothelial growth factor. This is a factor that stimulates blood vessel formation. We used a cardiac ligation model. So you give the animals an infarct. And here it's actually not VEGF. We made a Cree mRNA and put it into floxed stop lac-Z mice. So if Cree was expressed, the stop codon would be eliminated and you express lac-Z, which you could see with blue staining. And you could see in comparison to a DNA construct carrying Cree, a modified mRNA with Cree was dose dependent, very efficacious in the heart. Do a cross section. Actually, it's not just needle track. The needle track is somewhere here, but actually we got a very robust swathe of Cree expression is encoded by the modified RNA. And then we showed in a study that we published in Nature Biotech in 2013 that, this VEGF, when put into the hearts of these ligated hearts of these animals, could stimulate vessel formation and really help the heart regenerate post-infarct, as opposed to, not doing that, where you don't get vessel formation. So I put this slide together as an original sort of pitch deck for the foundation of modified RNA, just talking about the properties. So it's non-toxic. These are naturally occurring nucleosides. It's non-immunogenic. That's the key. It's dose titratable. You can control when expression. You can either make it transient, do one dose, or sustain, do repeat dosing. And it's incredibly versatile. I mean, we made literally hundreds of different modified RNAs encoding different proteins in the lab very easily, very fast. Took us about a week to make a new modified mRNA construct, purify it very rapidly, very cheaply. It's a nucleic acid, easy to synthesize, and therefore you could test therapeutic efficacy depending upon the target within a few months. This is disruptive compared to what normally happens with drug discovery. Use the patient's own cells to produce the protein, get the patient to produce their, properly translationally modified protein. You could make secreted factors via depot strategy, like I showed you with EPO, or you could, and this is unlike protein therapeutics, target intracellular proteins. And ultimately, I figured the cost of goods would be really favorable in comparison to protein manufacturing. So I founded Moderna in 2010, along with Bob Langer, primarily from MIT, long-time biotech entrepreneur. Just gonna check my time. And Ken Shen, who was an MD at Harvard at the time, now at Karolinska, originally raised only $2.5 million. It was the original site of Moderna. It's now moved, has multiple locations all over the city and now globally, quite frankly. So here's some quick facts. Since from the founding in 2010 to going public in 2018, Moderna was able to raise over $2.5 billion investment in partnerships. That's astronomical. But guess what? If you're trying to bring a new therapeutic paradigm to patients, it takes a lot of money. Company went public in 2018, raised a record $600 million ticket. Its now valuation is north of $60 billion, well over 1,000 employees. It's got a large GMP manufacturing facility about 20 minutes away from me in Norwood, Mass. 8 commercial subsidiaries expanding all over the world, as you can imagine, with this COVID vaccine that they've developed. And beyond that, there's 24 mRNA drugs making their way to the clinic, 13 already having entered clinical trials. So, the challenge we had when I first started Moderna was, mRNA makes protein, makes life, makes, potential therapy for whatever disease. So what to work on? There were so many possibilities, an abundance of riches. So now, 10 years later, the company has got programs going in all of these different areas: prophylactic vaccines, which you've heard about, cancer vaccines, immuno-oncology, regenerative medicine, secreted therapeutics, and intracellular therapeutics. So this is half of their pipeline. It's too, if I put it all into one slide, it would be too small to read. So everybody, who's kind of heard of Moderna recently thinks that this COVID vaccine is the first thing they did. Actually, not true. It's been in the clinic for many years now. Phase 2 for a cancer vaccine, phase 1 for a KRAS vaccine, phase 2 for another oncology application, phase 2 for, believe it or not, that VEGF application that I showed you about that we started in the lab many years ago, and multiple different programs on the way. Of course, the first product to reach commercialization is the COVID vaccine, which we'll talk more about in a moment. CMV vaccine is getting very close. Cytomegalovirus, phase 2, others in phase 1, Zika, They've now initiated a influenza program. They've already run rare strains of influenza, rare and deadly strains of influenza, but now they're going to do a seasonal influenza program. Nipah virus, we do not want to have a pandemic of Nipah. It's a very deadly virus, and other therapeutic targets. So I put these slides in yesterday because I figured everybody would want to hear about SARS-CoV-2, COVID-19, and the mRNA that Moderna developed for this, 1273. So as you know, the SARS-CoV-2 genome was published on the web by the Shanghai Consortia in January 2020. The next day, Moderna had an mRNA designed for the spike protein. 42 days later, it had manufactured GMP material that was ready for human trials. That is staggeringly unprecedented in terms of developing a new therapeutic. The timeline is crazy. But again, nucleic acid allows that. Same year, November 30th, results from the phase 3 blinded placebo-controlled trial were released. 15,000 received the vaccination, approximately 15,000 the placebo, and this was the results that really captivated the world. Day 1 injection, day 28 a second booster, and then this is the event rate of those that got COVID-19. So you can see that those that got the placebo, like everybody else in the population, continued to get COVID-19 at a pretty high rate because it was a raging pandemic, but those that got the vaccine did not. So it had a 94% efficacy. It was very well tolerated, no serious safety concerns. There was 196 cases in this study. 30 were very sick. They were all in the placebo arm. 1 death from a very sick person, again in the placebo arm. And so really it was 100% effective against severe disease. And it received emergency use authorization in the US and is now used in many countries around the planet. I get asked this a lot, why 2 doses and not 1? So actually in the same publication, New England Journal, they actually showed, even though the study wasn't designed this way, they showed the rate of the cumulative event rate from starting from it, first dose to second dose. And you can already see that by kind of midway through the first and second dose, there starts to be protection by being vaccinated, and even by this time point, there's seems to be very good protection. Not enough, not a large enough N to be able to say that, but it certainly looks that way, and that's consistent with how vaccines work. By the way, this is a booster, so there's evidence that a single shot will be efficacious. What about variants? This is something that I get asked all the time as well. So this variant was first identified, this B1351 was first identified in South Africa in October of last year. It's now in more than 24 countries. Convergent evolution, it's arisen again because it has a selective advantage. It has increased transmissibility. That's its selective advantage. And vaccines produced by Pfizer, Moderna, J&J, and AstraZeneca have all shown to be less effective, at least against this strain. And 2 trials run in South Africa, Johnson & Johnson and AZ, it's shown reduced efficacy in protection against COVID-19. Vaccines elicited by the standard vaccines were less effective at neutralizing this variant, although still pretty high neutralizing levels. So there is still protection from the initial vaccines. By the way, here are the mutations, and here's the spike protein here in red. So you can see this concentration of mutations, and these are probably the operative ones for making it more effective. But of course, the rest of the genome has mutations as well. So once this variant was looked like it was going to be a problem, Moderna made a variant-specific vaccine for it, and it was shipped, I think 3 days ago now, 2 or 3 days ago, I don't know what day it is today, to the NIH for clinical study. And they're going to do that in a number of ways, alone or in combination with the initial vaccine or as a booster. And these studies are planned. So, you know, the light at the end of the tunnel, vaccines are going to get us out of this. That's no question. You know, all the vaccines produced by, you know, we need billions of doses for the whole planet. So it requires all hands on deck, all companies on deck. But even for these variants that are emerging, people get very nervous about them. But you know, provided the fundamental biology doesn't change of the virus, and never say never in biology, but we can imagine vaccine strategies for these variants as well. And that's already started. So deep breath, drink of water, and I'll tell you about another story. So this is a story about improving the first stem cell therapy, which happens to be hematopoietic stem cell transplantation. So HSCs, hematopoietic stem cells, are the functional units of bone marrow transplantation procedures. HSCs first described in Toronto by Drs. Till and McCulloch, great Canadian scientists. So HSC transplant is, you know, in 50,000 patients per year across the globe at differing amounts. It's used to treat a variety of illnesses, primarily blood-borne leukemias, lymphomas, multiple myelomas, but other, you know, diseases, sickle cell, thalassemia, and other diseases can be treated as well. But despite this, it remains a very high-risk procedure, as bespoken by these numbers. The one-year survival rate for HLA sibling matched donors is only 70% and 55% for unrelated donors. Those aren't good numbers, so it's really a last resort therapy. What kills these patients? Well, it's primarily relapse if they're being treated for cancer, but opportunistic infection claims a number of patients, as does graft versus host disease and other things. So it's very successful. It was developed, HSC transplant, first successful transplant was done by E. Donald Thomas. And the longest surviving transplant recipient was this lady, Nancy McLean. I love this photo. This was a reuniting after 52 years. What I love about this photo, in addition to these two, is this person here. If you look closely, and I know you're all looking on iPads or phones or little screens, but you might notice a resemblance between the transplant recipient and this person. Well, that's because it's her identical twin was her donor. And to be honest, when this transplant was done, we didn't know about HLA matching, and this was really the only successful one you could do, was an identical match. But of course, then we learned about HLA matching, such that, you know, years later, you didn't need an identical twin. I love this photo too. This young woman had a very severe sickle cell disease, and she, Jocelyn Miller, and her donor was Petra Poker, a German lady, and it's just a wonderful photo. But like I said, it's really a last resort therapy. If you don't have to get a bone marrow transplant, your doctor shouldn't recommend one, because there's significant toxicities associated with it: severe infection, mucositis, you know, graft versus host disease, infection, organ failure. These are the things that kill you post-transplant. So really, even though there's a possibility for using HSC transplant for a wide variety of diseases, sort of the standard of care is here. It's about 50,000 patients per year. But there has been academic studies and trials run that suggest that you know, sickle cell, you can cure it with a transplant. Beta thal, you can cure it with a transplant. You know, as demonstrated on the last slide with that young woman who got a transplant for their sickle cell. But this just isn't done because it's too high risk, the procedure. There's also evidence that autoimmune diseases could be reset with a transplant, HIV, and others. Again, so there's a potential to transplant, you know, hundreds of thousands, if not millions of people a year, if it were a more efficacious and really safe procedure. So the process is such, you know, there's a donor, their stem cells are mobilized, taken out, they're processed, they're cryopreserved. And really the big thing is preparing the patient for the transplant. It's called conditioning. So you have to sort of clear out their old or their existing stem cells and often their cancer cells. So that's done with chemotherapy and high dose irradiation. And then the transplant is done. So we thought, well, there's a few places that you could intervene here, you know, improve mobilization. Some patients don't mobilize, or some donors don't mobilize very well. You could imagine expanding the stem cells. More is definitely better for HSC transplant. But the big one is conditioning. If there was ways that we could condition in a safe way, this could really open up the procedure. So this is what we sort of turned our attention to. And I just show you very brief data on two things that we did. One was we sort of developed a technology for expanding the HSCs ex vivo that was, and this is, people have been trying to do this for a long time. They typically go with a single agent strategy though. So for example, Michael Cook and others developed SR1, it's in the clinic. Guy-Sauvageau, UM171, but we took the approach that, well, you're not going to be able to get everything with one pathway or one compound. So we took a multi-pathway approach. This was the work of my grad student, Wataru Bina. He screened many combinatorial pathways and found that, and I'm sorry, this is coded, if you targeted 3 pathways, you could now get, you know, orders of magnitude stem cell expansion compared to what was out there. I think this is the big one though, which is the targeted conditioning. So we know that HSCs have epitopes on their surface that we could target them with, unique to them. So we envisioned immunotoxins attached to an antibody with antibodies specific for antigens on the cell surface of HSC. So we targeted c-KIT, which is expressed by hematopoietic stem cells. We hooked it up to a immunotoxin. When you don't condition animals, and this is mouse experiments, when you don't condition animals and you transplant them, you don't get any chimerism. You basically have to clear out the niche, clear out the existing cells to get the donor cells in. However, when we use this sort of antibody immunotoxin approach, first gave that to the animals, cleared out the stem cells, and then transplanted, we could get very, very high levels of chimerism, basically repair their blood. So studies describing this were published in Nature Biotech and Nature Communications a few years ago. And based on those data, I founded this company called Magenta Therapeutics along with my colleague David Skadden. This is an early picture of the initial team. Just some facts on Magenta. It went public. I think we founded it in 2016, 2015. Went public in 2018. It raised $100 million under its ticker. It now has a market capitalization of about $600 million. It's in the clinic, in phase 1 and phase 2 for multiple different indications. This is the sort of stem cell expansion product. This is the conditioning product. And this is the thing I'm most excited about, because if this works, this is really going to open up the field. So we wait and we hope. So again, a deep breath, and I'll tell you about one final story in gene editing. And it starts actually, at least from our end, with this fellow, Timothy Brown, the so-called Berlin patient, twice really unlucky and once very lucky. He was twice unlucky. First, he was HIV positive, and then he was unlucky again that he developed acute myeloid leukemia. But then he got lucky. He got transplanted by cells, and it was a designed experiment that had a mutation in the CCR5 gene. So CCR5 is the co-receptor for HIV-1 strains of HIV. It's really what's needed for those cells to, for the virus to get into cells. It had been discovered that patients with mutations in this gene, so-called Delta 32 mutation, were actually highly resistant to HIV infection. And so his transplant docs found a good match for him that happened to be from a patient that was Delta 32 homozygous mutant. So he was transplanted with these stem cells. It cured him of his AML, and he became the first person on planet Earth to be cured of HIV. So CRISPR, as you know, many of you, I'm sure all of you, was basically a bacterial immune system against bacteriophage. It made the jump into human cells in late 2013. And as soon as we saw that, we thought, well, let's apply to HSCs, our favorite cell, and let's go after CCR5 to knock it out, because we know that HSC biology is unaffected by deletion of CCR5, and there was this possibility of moving this towards the clinic. So we did this all in human cells. We took a two-guide approach. So you know that there's the Cas9 is the big protein encoded by the bacteria. It uses an RNA guide to go into where it then cuts. It's an endonuclease. So we use two guides to make a delta, to make a deletion in multiple different human donors. We analyze this as clonal level, because you can do this with HSCs, and we got actually very high efficacy of monoallelic or biallelic inactivation. Those cells still had the potential to be transplanted and function as stem cells. This is human cells into immunocompromised mice, giving chimerism, giving rise to myeloid cells and B cells and later T cells. And if you look at the bone marrow and spleen of those animals, the delta allele that we had created was engraftable. It was still in these animals. At the time, there was a lot of concern for the safety, you know, the on-target versus off-target of CRISPR. So we did a very, very deep targeted deep capture sequencing. We used, you know, we had 5 target sites. We used multiple guides and 126 off-target sites. And here's the on-target data first. It's a lot of data here, so I'll just draw your attention to this. We use single guides A, B, and C. We got, you know, on-target activity, low single numbers to teens. We used the double guide strategy to make deletions, and we got into the 20s and sometimes close to 50% efficacy of all the alleles were edited. And the off-target data was really amazing. So we were careful about our guides, and we looked at these 5 guides at a total. This is the total number of off-target sites, and you know, read at 100x coverage of the genome, and only detected 1 off-site event for guide B. If you look at guide B, it's not actually surprising. Lo and behold, it was supposed to be targeting CCR5, but it was also hitting the highly homologous CCR2. And this is the comparison between CCR5 and CCR2. So you can see that this guide is actually not a particularly great guide. It's totally, you know, homologous in the seed and the PAM of CCR2. So not surprising that this guide had an off target, whereas, you know, guides that had, you know, much more disparity in between their off-target site and their on-target site were much better. So this was the idea. You know, we take HSCs out, which you can do, mobilize them, take them out of a patient, treat them with CRISPR-Cas9, knock out CCR5, this is HIV patients, and then transplant them back in. And imagine, of course, if you could transplant them back into the own patient with a chemo regimen or a targeted antibody regimen that is well tolerated, this could really happen. So we got the cover of Cell Stem Cell, and I show this because it's kind of a cool cover, sort of targeting the HSCs very specifically. And this was published in November 6, 2014, and 12 days later on November 18th, Intellia Therapeutics, which was a CRISPR company that I co-founded, was announced its Series A. And this was in collaboration with some great CRISPR pioneers, Rudolf Barangu, Luciano Marafini, Eric Sonheimer, but you know, you'll probably recognize Jennifer Doudna, who's won the Nobel Prize for describing CRISPR with Emmanuel Charpentier. So this is where Intellia is now. It went public in May 2016. It raised $100 million. Now has a market cap of $3.8 billion. It's in the clinic. It's in clinic both by delivering CRISPR-Cas9 in vivo to patients for in vivo targets. Phase 1 studies have begun. It's also taking blood out. modifying it ex vivo, retransplanting back, as we've talked about. And what do you know, sickle cell is one of the targets here that has already started clinical studies. Interesting, you know, Cas9 is a protein. Guess what Cas9 is being expressed with by here? Modified mRNA. Not surprising. That's kind of cool. Two technologies in one here. So I'm going to stop there because I think my time is up. I could have told you about some other cool stories, but not enough time. Just to say that, you know, moving this, you know, basic discoveries out of the lab and into biotech really is the future of biomedicine. I think it's a really neat picture. This is Cambridge, Mass. I live in Boston, well, Newton, nearby, across the river. But this is a concentration of biotech companies. This is an old picture. There's actually many more of them now. Concentration of biotech companies here. Here's Moderna. Here's Intellia. Here's CRISPR and Editas. These are all CRISPR companies, incredibly. So, you know, I know there's a lot of students watching this. There is a future in this industry. But you may want to travel around the planet for a while, do some cool science, work with some smart people, and possibly start a family before you start your career in earnest. So I want to stop there and thank. This slide doesn't do really justice enough because there's so many more people involved, people in my lab shown here, collaborators, many collaborators, and associated with these various companies. But I will stop there, and I'm not sure, but if there's time, I'm happy to take questions.
Thank you so much, Dr. Rossi, for your remarks and insight on these amazing therapeutics and kind of the integration and innovative use of newer scientific technologies for the betterment of human society. This was a really amazing talk. Thank you once again.
Sure.
You are correct. We do have a couple of minutes, around 10 minutes for questions. So attendees, feel free to use the chat or the Q&A feature to ask Dr. Rossi any questions. And if you wish to address him directly, you can use the raise your hand feature at the bottom of your screen, and then we'll enable you to unmute yourself. So I do see one question in the Q&A, so I will ask you that, Dr. Rossi. The question is by John, and he says, are the vaccines being developed for cancer preventative, like the flu shot, or restorative, like that for rabies, or therapeutic, like a vaccine substitute for chemotherapy?
No. So, well, just to make sure I understand the question, because it was complicated and my head's, you know, I haven't had another cup of coffee. The idea is to, the intratumoral vaccines for cancer are to elicit largely immune attack of cancer cells. So some using neoantigens. This is personalized cancer vaccines. So a lot of tumors make neoantigens. And if you could present those neoantigens to immune cells, you could hopefully stimulate them to attack cells expressing these neoantigens, cancer cells. So that's one approach. The other approach is just molecules that will, you know, tumors are very good at creating signals that prevent the body's recognition of their cells and to, you know, have your body come in and attack the cells. So some of these strategies are designed to elicit immunosignals within the tumor to bring in the immune cells. And there's other strategies as well. I'm not sure that quite answers your question because I'm not sure I quite understood it. My apologies, but there's a few different strategies ongoing in the cancer vaccine space.
Thank you for that. And John actually clarified his question a bit more. So now he says he's pretty much trying to ask, will everyone eventually get these, or are they for cancer patients? I'm assuming.
No, I don't think, you know, it's very hard to, thanks for the clarification. It's really hard to, although we do it with infectious agents, to you know, medically treat people that aren't sick. You know, vaccines are one of the rare instances that we actually do that. But you know, we've been using vaccines as humans for over 500 years, and they've proven their safety and efficacy. I would tell that to all the anti-vaxxers out there. I'm sure not many of them are at this conference. But you know, when was the last time you saw somebody walking around with polio or smallpox? I mean, vaccines work. Though prophylactic vaccines, for running trials for rare diseases like cancer that come up, that may come up, and there's so many different types of cancers. I think it would be a tough sell for the FDA and for running clinical trials, unless of course the safety, after years is so efficacious. And you think about P53 mutations, it's mutated in most cancers, KRAS. There might come a time, so never say never, but I don't think we're going to see it as a prophylactic measure. Rather, it's going to be in people that have cancer. These are certainly the first clinical trials that are being run.
Thank you for that, Dr. Rossi, and John also thanks you for your answer. Now, Usman is asking, although you have retired from academia now, what are some gaps in the hematopoietic field you would recommend future researchers to look at?
Well, the fundamental mechanisms controlling their self-renewal are still poorly understood. We heard a little bit from Dr. Kaplan. I caught the end of his talk talking about the niche and how the niche nurtures the HSC. Mostly, it's amazing. I started off asking fundamental biology questions, but then after I sort of caught the biotech bug and moving discoveries towards a clinic, I typically think about things in the setting of, if we were to answer a question, could it move towards patients? So just imagine all the blood disease that's out there. Whenever it's blood-based, imagine asking questions related to, what if we answer this question? Could we solve that? That's true of all blood-based diseases. But blood is, I kind of refer to it as the coral reef of the body. It impacts every one of our tissues. So I would say that you could really think, and if you really wanted to think out of the box, to say, well, how if I do something and achieve a goal in blood cell biology that we can do, let's say by transplant or various other strategies, how might that impact other aspects of human disease? That's where I'd be thinking. I'm not going to give you specific ideas per se, but I would encourage you to think along those lines.
Derrick, it's Arnold Kaplan. I'd love to ask you a question. Since I'm so old, I want a magic elixir for anti-aging, and what we know is that the load of senescent cells actually has a huge influence on organ malfunction and in the aging process. So do you think we could have an anti-aging vaccine that went after senescent cells?
Yeah, it's an interesting idea. I studied aging, as you know, Arnold, for many years, and you may have noticed that none of the companies that I started actually had anything to do with aging. And I hate to be a downer here, but we age really well, really so completely. So much goes wrong, and it really comes down to our evolution. 1,000 years ago, the old man in the tribe would have been 35, 40 years old at most. We never even had a chance to evolve beyond 50, 60, 70. So we're living in some sort of strange evolutionary time now with modern medicine that we're living out well beyond certainly our reproductive lifespan, but even our evolutionary lifespan. And we do really fall apart so incredibly. Our cognition declines, our immune function declines, our tissue homeostatic ability declines, our DNA gets mutated. So of course we develop, tumors here and there, and if not tumors, our cellular function declines. So I think that, and this, increasing lifespan, do we really need to live more than we live now, 80, 90, 100 years old? Health span, I think, is something that people are interested in. So, and is something we could approach. So if you imagine health span, you can imagine targeting a particular mechanism. For example, like you said, senescent cells. That is approachable. That's probably not going to give us lifespan because of the rest of us is falling apart so completely, but it could if it's targeted, let's say, to a certain tissue, or maybe not, maybe multiple tissues, we could imagine increasing health span. I never found a really good idea in the research we did on aging, which was for a long time was actually central focus of my lab, but something that I felt had the legs to move into the clinic, much to my chagrin.
Thank you for that. Dr. Kaplan, do you have any further remarks or questions?
Yeah, I would just tell Derrick that there's a phase 2 clinical trial using MSCs as an anti-aging elixir. And the logic for that is that there's a geriatric scoring chart for active aging, and this multifunctional scoring system can be evaluated. So I think on the aging end, I agree with everything you just said quite wisely, but having developed a scoring system for severe aging and showing that you can improve the scoring system is actually a therapeutic mechanism. That same but different scoring system is being used for clinical trials in autism. So I think the issue for me to get something to a clinic is, as you know, is to identify outcomes that can be quantitated.
Absolutely.
And that's the key, I think, for health span, is to identify scoring systems that we can rely on to help improve. And so, Derrick, hurry up and get something that's anti-aging. I need you.
Well, me too. Arnold, that I working with some of your case colleagues on another company, which I didn't get to talk about, but with Paul Tazar and Drew Adams with Convello, which is a wonderful story, which I didn't get to tell you all about today, but I'm sure you know it well.
For sure, and Paul Tazar is a great story, just like yours. He was an undergraduate at Case Western Reserve and had a fabulous PhD program, and we recruited him back. So I think for the students who are listening, they can do the Derrick Rose approach, or they can do the Paul Tazar approach, which is get really good postdoctoral experience in a number of laboratories, and then come back and set up your own lab. And actually, your entrepreneurial activity is clearly dependent on how many transatlantic flights you can make.
Thank you so much, Dr. Kaplan, for your question and also for your advice. Looking at the time, I think we have time for one last question for you, Dr. Rossi. So, anonymous attendee actually asks, are there strategies to reduce the currently prohibitive cost of stem cell therapy?
Well, so manufacturing is something I think a lot about because it's a manufacturing issue. Cell-based therapy is really expensive, particularly if it's on a patient's specific level. So the manufacturing costs associated with that are astronomical. One strategy that people are taking, though, to try to surmount that is, instead of having, every patient get and I know MSCs are immunoprotective, so that's excellent, but most cell types you can't just transplant from one patient to another because they'll reject them. But there are many, and in fact, now I'm sitting on the scientific advisory board of a newly formed one, companies with the effort, with an effort to make a universal donor stem cell, so universal iPS cell, for example, that, and it's, what do you know, involves a whole bunch of CRISPR knocking out and modifying a whole bunch of different genes that are involved in when you transplant a cell that's not self being recognized as not self, and rejection resulting. So I think actually that is a possibility, because now if you have, when that succeeds, a universal donor, now you can large scale, there's always economy of scale. And if you could, if it's just one donor, you can do that in large scale. That'll really reduce the cost towards getting therapies derived from those universe. And that's, you start with iPS cells, then you have to make, the cell type that you need. But I think that's, specifically to the question that you asked, are there ways to lower the cost of this? I think that's a future thing that's going to happen. I know people are working on it.
That's an excellent perspective, and something that's pretty new to me, at least. So thank you once again for that. And thank you for your time and your amazing talk, Dr. Rossi, as well as Dr. Kaplan, who was here previously. Dr. Rossi, we will now redesignate you back to an attendee, and we hope you enjoy the rest of the conference and continue to participate. Thank you so much for being here.
Thank you all.
So this brings us to the end of the keynote speech, as well as the opening ceremonies. The research and bioethics case competitions will now take place in the research and bioethics halls respectively. You can find the link to these sessions in the attendee booklets. All competitors must be present in their respective halls, and we highly encourage all other attendees to participate as well. And we hope that all our attendees have an amazing day full of fascinating science, bioethics, and business. And I believe the bioethics hall link will shortly be shared in the chat as well for those who cannot access the attendee booklet immediately. Thank you once again.
Hi, everyone. So I hope you enjoyed the opening presentation and keynote presentation.
Oh, I'm sorry. That is my bad.
No worries, no worries. So firstly, we apologize about running a little